Well, everyone, welcome back to another Expert Talk series. There was a word in the title called crowdsourcing. And to me, when I saw that, I knew that it was us because we are the crowd who's going to come up with the source that's going to change this whole program. So I just want to welcome each of you here, and always remember that our library is full of all of these wonderful talks. And with that, I'm going to want to turn it over to our CEO and founder, Indu Navar.
Thank you, everybody, and for joining us today. I would love to actually introduce Dr. John Landers, and we are honored to have John here and present to all of you today on crowdsourcing and accelerating, genetic findings through what ALS compute project that he's been working on. And John Landers' PhD is an international leader in the field of ALS genetic research. He received his PhD in molecular biology from University of Pennsylvania. Dr. Landers was then a postdoc fellow in genetics at MIT. He was also a chief scientific officer at Polygenics from 1999 to 2005. And he's also taught at MGH and Harvard Medical School. After that, he began as an associate professor of neurology at UMass Chan Medical School. His career has been mainly focused on using new high-throughput technologies for the identification of neurodegenerative disease gene with a strong focus on ALS and FTD. Through his efforts, Landers' lab was developed extensive collaboration with leaders in ALS and FTD genetic research. The Landers lab as major contribution to identification of FUS gene that most of us know here as a significant percentage of familial ALS cases. Through the long-standing interest of novel technologies, his lab focused on new next-generation sequencing approaches to identify additional genes contributing to ALS and FTD. And this includes using whole genome sequencing, RNA-seq, and exome capture sequencing. If you don't know any of these words, it's okay. John Landers is going to educate us. This effort has led to identification of PFN1 mutation in familial ALS. Most recently, the lab has focused on using rare variant analysis of exome sequencing results to identify novel causative genes for ALS and FTD. Through this effort, we have, he has shown that mutation in TUBA4A gene, further optimization of rare variant approach through the use of training data set to discover NEK1 and recently KIF5A as ALS and FTD associated genes. With that, I would love to invite Dr. Landers. As you've seen, he's been busy working through the genes that a lot of you have actually contributed to and continue to contribute. So Dr. Landers, the floor is yours.
Thank you for that wonderful introduction and the invitation to be here. Before I start, I just wanted to let everyone know I have a major storm going out my window. So if I lose power and disappear, it's not because I don't like anyone on the call here. It's for other reasons. So let me get started in talking about ALS genetics and kind of our efforts with the ALS Compute project and how we want to use this for crowdsourcing purposes. So to start off, what I want to do is give you a little bit of an idea of, why are we interested in genetics? And the main reason for this is because genetics does open up several avenues of research for therapeutic treatments. As we find genes, they can lead us on, different routes to get to that therapy. It can give us information on what pathways or what mechanisms or what things are going wrong within ALS patients. It can help us develop model organisms such as mice or even worms and zebrafish to kind of mimic ALS. It can help us to classify patients as well. and that, if we know a certain patient has a certain mutation, the therapeutic for that may be different than someone else. And then much more recently is the application of gene therapy, where if, again, more prone towards familial ALS, but if we're able to actually activate a mutation, that can lead to that is a direct therapeutic treatment. So all of these methods all stem from genetics and will help facilitate the development of these therapeutic treatments. So to take this a little bit further, we can also break down all of ALS into two categories based on genetics. And I'm sure most people on the call are familiar with both of these groups. First, we have familial ALS, or FALS as we call them for short. These are patients that have a family history of ALS, and they represent about 10% of all ALS cases. Typically, in these patients, the ALS is due to a single causative mutation in a single gene. To date, researchers have identified about 2/3 of all the genes that are contributing to familial ALS. And quite often the way we think about familial ALS is that it has a very, very strong genetic component to it and a much, much weaker environmental influence to that. Now, in contrast, we have sporadic ALS, where you do not see any family history of ALS. This represents about 90% of the cases. And quite often the way we view sporadic ALS is that it's not just one gene contributing, but many different genes that may be contributing to your risk of ALS, plus possibly environmental factors. Up to date, there's been very few risk genes that have been identified which contribute to ALS. And so you can think of this as kind of the reverse of familial ALS, where it has a weaker genetic component, but probably a stronger environmental influence to it. So to help you understand a little bit of the talk and what we're doing in genetics, I want to give you just a little bit of background on human genetics itself. The first thing is knowing about the human genome. The way you can kind of think about the human genome is it's one very, very, very long word, a word that has about 3 billion letters in it, except there's only 4 letters in it.
It's just, it leaves me in awe.
A, C, G, and T. Those are 4 letters. And within the human genome, what we usually find is that there's about 25,000 genes within the human genome. And each one of these genes essentially contains the instructions to make a single protein, which will be used in the cell. And what's kind of interesting is when the Human Genome Project was accomplished, it was a little surprising that on how similar all of us are. If you take any 2 individuals on this call, they're about 99.9% identical on the DNA level. So next time you're at work and you're looking at the person next to you, you can think to yourself, hey, I'm very similar to that person. You may like that, you may not like it. The other way you may think about this, though, is that, yes, I'm very similar, but I also have 3 million differences between myself and the person next to me. Human genome sequencing has been just massively going on over the past 10, 20 years now, and over 380 million DNA variants have been identified to date. Now, most of these are benign. They really have no effect whatsoever, but there are variants or mutations in some genes that can cause human diseases like ALS. So one of the questions that comes up is, how can we identify causative ALS genes when there's so many variations in the human genome that are benign? How do you pick that needle out of the haystack? And the way we actually do this, and it varies between situation to situation, is that we essentially compare the human genome of people that have ALS to those who don't have ALS, or as we call it, cases versus controls. Now, as I said, there are millions of variations that occur in the human genome. So in order for us to really get to the point where we feel confident in that something is more abundant in people that have ALS versus controls, this may require us to do thousands or tens of thousands of genomes for that comparison. One very overused analogy that I give to people is that, if you were trying to identify a single mutation, this is essentially equivalent to me coming into your office, putting down 5,000 copies of War and Peace and saying there's one typo in all of these books, go find it. So it is essentially a needle in a haystack process that we are going through. Now, there's been tremendous advances in DNA sequencing over the past 1 or 2 decades. And as a result of this, we've identified, as the scientific community, numerous familiar ALS genes. And here's a list of several of them on the left, and you can see the year discovered and the kind of the percent of familial ALS cases that you see mutations in this particular gene. So as you might expect, C9orf72 is high up there on the list. SOD1, TDP43, and in FUS, and NEC1 and TPK. Those are the highest ones that are out there. Now, one question that I often get asked is, what's the purpose in discovering these genes that have low frequencies in the ALS population? Why do you want to find a PF1 or profilin-1 or VCP or one of these low ones? And second is, shouldn't our research actually focus on sporadic ALS, which accounts for 90% of all ALS cases? And I want to address, kind of both of those questions. And if you don't understand the answers to this, what you really have to do is understand what is going on within a neuron within ALS patients. Within the picture that you're looking at, you're going to see a very large list of all the different things, I should say all of them, a portion of all the things that are going wrong within the neurons of ALS patients. This is a very, very small number relative to the total number. And because of this, if you're looking at all these different defects that are going on and you're a drug company, you're going to say to yourself, well, which one should I focus on? This one over here or that one over there. And as a result of that, that really complicates where, if you're developing a therapeutic, where you want to focus your effort. Now, where genetics comes into play, though, is it can actually pick out those primary pathways that are defective in ALS patients. And the way we can actually do this is if you look at all the familial ALS genes, they're, for the most part, all broken down into 3 pathway categories. We know that there's an issue with protein degradation in ALS patients. We know there's issues with RNA processing, and we know there's problems with the cytoskeleton, which is essentially a structure that gives rigidity and also transport molecules within the neuron. So just by familial ALS genetics, we can see the primary pathways that are going wrong. Now, if we did not study these very, very low frequency mutated genes, we wouldn't be able to get to this point. We wouldn't be able to figure out which pathways are probably the highest in terms of contributing to ALS. And the other thing to keep in mind is that the defects that you see in a familial ALS patient versus a sporadic ALS patient are extremely similar to one another. And therefore, knowing these pathways, we can start to think about therapies for not just familial ALS, but sporadic ALS. So most recently, there's been numerous obstacles that have been limiting our future discoveries in ALS. We are getting to the point we have found lots of familial ALS genes. We do want to focus more efforts on sporadic ALS, and again, the advances in whole genome sequencing have permitted larger and larger studies. Now, I can tell you that there are numerous ongoing ALS whole genome studies that are active right now. I'm sure you probably know at least some of these, the Answer ALS project, the CREATE Consortium, the GTEC project, New York Genome Center's ALS Consortium, NIH, and Project Mine. Now, it's great having all of these different efforts. However, it also does create certain problems. And one of the things that you have to realize is when you're doing whole genome sequencing, it really, really requires you to have some high performance computing. This isn't something you can do on a laptop. You need massive computing, you need massive storage of your computer files. And as a result of that, it's limited in terms of the number of people that can accomplish this. Also, if you're looking at this list, you may say to yourself, well, they're all doing something similar. Why can't they just kind of, there's probably a duplication of effort, and as a result of that, there's a loss of cost and time and labor. Another thing is there's a lack of standardization in the field. In other words, if Answer ALS is analyzing their genomes one way, in all likelihood, the other groups on that list are analyzing them a slightly different way. And as a result of that, it makes it difficult to actually combine data from these different groups. It can't be where we just call them up on the phone and say, can you give me all the variants and send them over, because they were identified by different methods.
I know.
And so, the bottom line for all this is that due to the high cost of computing and the limited availability of data, this really restricts the number of research that can study ALS genetics. So, this is something that I had discussed with two of my colleagues, Brian Traynor and Jonathan Glass, who some of you may know. And, we took a look at, these problems, and we said, is there a solution to this? And from this is where we came up with the idea of the ALS Compute project, where essentially we want to create an open central repository of ALS whole genome sequencing data. So in essence, our goal is to take all of the whole genome sequencing data that exists in the United States and Canada, consolidate it, bring it all together, analyze it from scratch, and then put all that data up in the cloud and make that data as accessible as possible to as many people as possible. So by doing this, we get around many of those problems. So first of all, by consolidating data, you get an increase in the quality of the genetic studies, because now we do have a standardization of how we process all the DNA. The quality of the variants and mutations that we identified is much higher. We're reducing costs because now we're not all doing the same thing. And as a result of this, we're hopefully increasing the number of novel genetic findings that are going to occur in ALS. Also, by putting this up into the cloud, we're also providing everyone all the necessary computing and storage infrastructure that you need. The initial version of this, which I'll get into in a minute, was put up on Google Cloud. And so if you want to use it, you just need, frankly, a Gmail account and go through a process of getting access to it. But once you have that, you can use all of Google's computing to analyze the whole genome sequencing that is present within our project. And so as a result of this, our feeling is this is going to allow all researchers the opportunity to study ALS genetics. And, we want people that are outside of ALS to actually study this. We do want Parkinson's experts, Alzheimer's experts in genetics in general, to all study ALS genetics. We want to bring them into the party. In addition, what we really want to do is bring in experts in other fields. And what I mean by this is that there is, as genomics gets bigger and bigger and bigger and bigger, how we analyze genetic data when I learned how to do it is totally different than how we're going to be doing it in the future. In many respects, I'm starting to become obsolete. And what we want is to get people that are experts in big data to be analyzing these, people that are experts in machine learning and neural networks and, dealing with just big data, that to look at this and to say, is there something that you can pick out that we can't pick out? So we start the ALS Compute project, and what we initially did is went to all of these large sequencing consortiums that I mentioned. Addition, we also included Canada as well. And right off the bat, every single one of them said, yes, we're more than happy to contribute our data. We're willing to share it, take it, let's harmonize it together, make it available to as many people as possible. So the members of each one of these projects should be applauded for their willing to collaborate on this project. We initially put out a set of about 7,400 genomes back in July, and that was produced by combining sequencing data that I have with Brian Traynor. And you can see it's about 7,400, and you can also see the breakdown of ALS versus control versus other samples that are in this whole genome sequencing. And this, like I said, was released on Google Cloud. In about 2 months, we're going to be releasing what we call our next freeze, and this is going to increase the number of whole genomes to about 12,600, and increasing the number of ALS FTD cases to about 7,600. Due to situations beyond our control, we are switching from Google Cloud to Microsoft Azure's platform, but essentially the concept is the same in that it is within the cloud, and you'll have access to all the tools necessary to use that data. And we're also starting to plan out for the next release beyond this, which we anticipate will be in December. We've already received about 3,000 more samples, and we've processed many of these already. And we anticipate that this number will go up several thousand as well. So all this data is, or at least for Freeze 2, is about to become available to anyone that wants to get access to it. The way you actually get access to this data, we unfortunately can't just put it out on the internet. What we have to do is essentially release it through NIH, and they have a database called dbGaP, and all you have to do is go on there, say, I want access to the data. They'll ask you a few questions in terms of what are you using the data for? Are you going to do anything nefarious with this data? And once you do that, in about 2 weeks, your application will be approved, and you'll have access to this data. So once that approval goes through, you can get right on to either Google or Azure, depending on the time frame, and get access to all that whole genome sequencing data. And what is nice is the way that we've actually stored this data is that we've made it available such that you can use numerous tools that are available for genetic analyses. You're probably not familiar with some of these, Terra or Jupyter Notebooks or Bioconductor, but all of these are used commonly in genomics to analyze genetic data. And given that, you can access all of this data directly within these programs. In addition, there are a number of very, very, very large data sets out there available that you can incorporate into your analysis. MPD is one that you may be familiar with, or TopMed. And so it makes it a lot easier, since it is all in the cloud, to incorporate all that data into your analysis of the ALS genomes. The type of data that we provide is listed here. We give people a number of different files. I don't expect you know what these are, but in essence, what we're giving people is the raw data, unprocessed, right off the sequencing machine. Another version where we've done some initial processing, and then another version where we go through the full processing to give what we think is the highest quality data possible. In addition, we give anyone who gets access to this a phenotype file, which has as much clinical information we have on the patients that are within this cohort of whole genome sequencing data. And again, we're trying to make everything as easy as possible for the user to do any type of analyses they want. One question I get asked is, well, can I download this? And yes, you can. One thing you should recognize, though, is if you want to download all of this, it's about 252 terabytes of data, and Google charges you a fee to download. And to download this much data will cost you about $15,000. And also, just to put this in perspective, your typical laptop has about 1 terabyte of data, or it could store about that much. So, the idea of having this in the cloud is to get away from people downloading this and have everything much more easily accessible to everyone. As I mentioned, we go through a processing step, initial processing, and then a higher level of processing. And this just gives you an outline of the steps. And again, I don't expect you to understand the steps in that we're doing here. But the bottom line is that after doing all this, we end up with about 264 million variants identified in over 13,000 whole genome samples. Or if you multiply these together, you get about 3.5 times 10 to the 12th genotypes that we are providing to people. Again, to bring up an analogy, just to give you an idea on how big this set is, 3 times 10 to the 12th is about the number of seconds that occurs in 112 or 113,000 years. So just to give you an idea of the magnitude of the data. One of the things that we're also providing to people is the ancestry of each one of these samples. If anyone here has done, say, 23andMe, the way they do it is pretty much identical to the way that we do it. And it's essentially just looking at different variants in the genome and how similar they are to other different ancestries. Probably the one big thing to point out is that a large majority of the samples that we're dealing with are of European descent, nearly 90% of these. And, I mainly point this out just to bring out that there is a strong need to collect more ALS samples from these other ethnic backgrounds. There are some projects out there that are doing this, but frankly, they need more money, and anything that we can do to get more populations into these type of studies would be tremendous. The other thing that we provide to the user is that we look at many different genes throughout the genome and determine if they have what's called a repeat expansion. I'm sure many of you are familiar with C9orf72, and that there is essentially a sequence of DNA that ends up repeating itself over and over and over again to very large sizes. And that's what the cause of contributing to the development of ALS. We've actually taken about 30 of these similar type diseases, such as myotonic dystrophy or Huntington's, and have gone through and analyzed each one of these genes so you can quickly determine who has an expansion and who doesn't. So for instance, on the right here, you can see a column here for C9orf72, and you'll see 2 people highlighted that have, the first one here has 208 repeats, the other one has 320. These are very, very, very large compared to all the others. These are 2 patients that have an expansion in C9orf72. So by providing this information, the user doesn't have to do this step. They have access to it. We are also going through and defining all the possible mutations that may be present within this ALS cohort. I'm giving you kind of a breakdown here where we have about 818 C9 patients, 246 SOD1, 96 TDP43, 34 FUS, 52 NEC1, and 14 TPK. Again, if it's something where you're looking at, say, sporadic ALS versus familial ALS, it could be that you want to not incorporate these samples into your analysis. Or let's suppose you're interested in something about C9, you're going to limit your analysis to just those 818 people. So again, we're trying to do the upfront analysis for the user, do as much as possible so they don't have to do it. In addition, every single variant in the genome can be described by many, many, many different programs that are out there. I'm giving you on the screen now just a list of some of these programs. And what we've done is analyze every variant that we've identified with every one of these programs. And again, this is again to take the burden off the user and give them that data so they can advance with their findings as quick as possible. We've also incorporated some tools for more advanced bioinformatics people. In essence, what we've done is taking all the genomic data and actually put it into a database called HALE, which is specific for very large data sets. If you're familiar with the All of Us project or the TopMed project, they both use this database for handling all of their data. We're providing all of this information to the user. If you're an advanced user, that's great. In addition, we provide all the annotations you see in the box on the right to people that are able to use the data in this particular format. Now, we're also we recognize that most people out there are not bioinformatic experts out there. And so we're also trying to make tools to make it as simple as possible for you to analyze this data set. I'll give you one example here where let's suppose you wanted to pull out all of the mutations in the SOD1 gene. This is literally the code that you see in this box here to do that. And if you do that, the output you see is on the bottom, and it'll give you for every single mutation that it finds, what amino acid is changing, how many people have that mutation, and on the right here, what samples have that mutation. If you don't want to do SOD1 and you want to do FUS or TDP43, just cross out SOD1 and put one of those genes in there. Similarly, we're adding a similar type of code for you to do a number of other different analyses with having little to no experience in bioinformatics, so you can use this database as well. Up to this point, I've been talking about whole genome sequencing data and how we're trying to make it the best quality as possible. But one thing I haven't touched upon is the clinical data. And one of the issues that we find on my side is that as we get clinical data or phenotypic data from different groups, how different clinicians kind of measure different clinical features is done differently. And as a result of that, if you're getting data from one person, one clinician, it may be different than another clinician. And so as a result of that, what we have done is kind of called together a committee of some of the top ALS people in the world to come together and say, can we come to a consensus? If we're saying, age of onset, how are you actually defining that? What is, the key traits? If we're measuring progression, when do you measure when progression ends? What's the exact date that we can do this? And as a result of that, their goal is to come up with a data dictionary for all of the ALS-related clinical phenotypes. And so lastly, I just want to go back to my original title, which is using all this data for crowdsourcing. As I said, we're putting it out there. We want people to use it. We want people outside of ALS to use it. And so how do we do that? And, we're doing this mainly through setting up crowdsourcing competitions. And again, this is, with your group, Everything ALS. We're working with your group to develop one of these challenges, the End ALS 2.0 challenge. It is being helped out by Roche Canada as well. The idea is, let's put it out there. We'll come up with a certain question that the general population needs to answer, and whoever does it best gets a cash prize. We are holding this through Kaggle. If you're not familiar with Kaggle, they hold a lot of these type of competitions, and we're hoping to have this set up by June of this year. We are also in talks with other groups as well to have competitions. One of the other ones that we're talking to is the Challenge Prize on ALS, which comes out of the UK's innovation agency called Nesta. And this type of challenge is more geared towards large teams or companies. This is where your Googles and Microsofts and what have you may be participating. And the incentive here is that it can lead to very large prizes, somewhere in the order of 250,000 to 2 million pounds. They've held 80 competitions in the last decade, and hopefully we're going to be able to use this data for their next competition. So with that, as you might imagine, there's tons of people to thank. I obviously won't go through all of them, but obviously, if you know any people from any of the projects I've mentioned here, you should thank them for their contribution. And I will stop there and take any questions.
Perfect. Thank you so much, Dr. Landers. And we've got a ton of questions, and Martha Beach and myself, we are going to be moderating. So let me kick off with the first question we have is, do variants of unknown significance in ALS genes affect the rate of progression of sporadic ALS when present?
So I guess first I have to address the variant of unknown significance. So presumably that was derived from a DNA diagnostic test from GeneDx or one of the other companies out there. And when you see that, what it first of all says is that the company itself, GeneDx, for instance, is unsure whether the variant they see is a mutation or not. Now, without causing too much, controversy, the scientific community does not always agree with GeneDx. And what they call a mutation may not be, and vice versa. I've seen variants of unknown significance, especially in the NEC1 gene, which we work on, that I would totally disagree with and say they are true mutations. So you have that aspect off the bat. In terms of, but now that you know that, to understand the progression part of that question, it's more important to understand, is the variant they're looking at a true mutation, or is it, say, I don't know, I'll call it a minor risk factor, or is it, in a gene that has a very fast progression? If you think of A5V within SOD1, that is a mutation we know has a very fast progression. And so it's not so much the variant of unknown significance, but rather what is the mutation itself and what gene it's in. Probably one more thing to add to that is, getting an idea of progression from a particular mutation is very difficult, given that C9 and SOD1 are the most, prevalent ALS genes out there. Some work has been done with those, but anything below that, except for a couple of rare examples, there's not enough data to really say one way or another whether it's a short progressor or a long progressor.
Thank you.
Along that same line, we have a question about in familial ALS patients, do we find the same gene is always, or sometimes usually, the cause of the disease within that family?
Nearly all the time. And the reason for that, and actually one of the reasons that we've actually been able to identify these genes is because they are within a family. We can look at the different family members and start to see what DNA is in common between all of them. As you know, every one of us is derived from our mother and our father. And so, and you share about 50% of your DNA with any sibling you have. And so if you're looking at, cousins or even more distant relatives that have ALS, what you want to see is, what is in common between what they have and what you have. And by doing that, what we are able to do is get it down to a small region and identify what those mutations are. So in essence, in nearly all the cases, it is that the same mutation is occurring within all members that are affected within a family. There are rare occasions where we've seen it doesn't fit that, and should also keep in mind, we look at thousands of these, and I can just quote you a handful of it that have occurred like that. I've seen one where C9 patient has relatives that don't have the C9 expansion, is one example. Probably more common, I would say, is or more realistic, not that they have a different mutation, but you see a sporadic case within a family of ALS cases.
Mm.
So in other words, they may not have the mutation the rest of the family has, but they're developing ALS just because they're essentially a sporadic ALS case. And so looking at that, you look at it and you say, well, they don't have the mutation. It's just they're getting it just as a sporadic versus familial ALS case.
Very interesting. One more component to this question, Dr. Landers, from our group. How far into the family tree, when you're looking at familial ALS, how far into it are you examining the family tree?
From my point of view, my goal is to identify as many genes as possible. And in genetics, one of the great things about it is the more the better. It's kind of the slogan for it. So if we can get our hands on as many family members as possible, we go for it. Now, I'm not a clinician. I don't deal directly with patients. Quite often I'm dealing with clinicians that are collecting the DNA from families. But each one of those knows enough to go out and get as many family members as possible and to bring them together for us to analyze.
Thank you.
So there's a question on collaboration. If someone is in ARC, which is part of ALS TDI, or a platform trial, or even CDC, will their data be included in Compute ALS?
That's a multi-tiered answer.
Yeah.
Let me start with, it depends on the group itself. So ALS TDI has recently contacted me to say they would like to contribute their samples, whole genome samples, to this project. Having said that, my understanding is they're also contributing them to Answer ALS as well. In terms of the clinical studies, no, it's not the case. And this is something that I would love to get changed. In my personal opinion, when groups are holding clinical trials, one thing they don't do that they should is, frankly, to do whole genome sequencing on those patients. And that could be very, very useful in determining whether a drug is being effective or not. It's already known that each of us metabolizes drugs in different ways. We know that there's other drugs out there that some people can take and some people can't. And so if you're not looking at the genomic DNA in your clinical trial, to me, I think you're not doing it the best you could. I would love to be able to do this. The feedback I've gotten, though, is that some of the drug companies don't want to participate. So if you would like to call them and push them, you're the perfect group to do it.
Perfect. Thank you.
Thank you, Dr. Landers. We have a question about, can you develop a quote antidote customized for each patient to address the malfunctioning gene? If so, how long would it take? I apologize. How long would that, and would you anticipate that it would bring that to market?
Sure. So if we're talking specifically about familial ALS mutations, it will obviously depend on the gene itself. Obviously, SOD1 with tofersen and with FUS, we've already seen advances in developing these drugs. For C9, the trials didn't obviously work very well. I can tell you, though, that my lab, and I know numerous other labs, are addressing each one of these genes. And to be able to just frankly go down the list and say, all right, we can develop something that can inactivate the mutation somehow, some way. Now, once that's something we can do very easily within cells, the neurons that we grow within the lab. Depending on the situation, we might be able to test it in mice. Beyond that, though, I'd say the biggest limitation is the getting to that phase 1 clinical trial. Clinical trials are not expensive or not cheap. They are very, very, very expensive. So it's kind of a combination of how far do we need to develop something, and can we raise enough interest to take it to the next level to get it to a clinical trial. And hopefully over time, some of the more rare mutations will be addressed. And we're trying to do that, but it does come down to a little bit in terms of money as well. I know you don't want to hear that, but that is the reality of the situation.
Yes. Thank you.
Yeah. So there is a question on, will ALS Compute Project offer opportunities to identify new gene mutations? Is there a focus on recruiting people with familial ALS, but whose genetic testing shows no known mutation?
Absolutely. We want every single ALS patient we can get our hands on. And if it's a familial ALS case where the mutation hasn't been identified, there's definitely going to be a focus on that. There are other methods that we can use. Just one example that comes to mind is even within sporadic cases, there may be individuals that are related to one another but don't know it. And so what we can actually do is actually determine within that group who's related to everyone else. And if we see two people that have ALS that are within the same family, we're going to focus on those as well. And I've had similar situations like this before. I had one case where there was two brothers, each being seen by different clinicians, one down in Atlanta, one in Boston. And these were brothers that both had ALS and hadn't talked to each other in about 15, 20 years. But through our analysis, we were actually able to determine that they were brothers. And that is something that we can focus on then and say, all right, this is a familial ALS case. How can we use this to our advantage to identify a new gene?
Thank you.
Thank you. And then we have another question. Why is Compute Project only here in North America, and will access Project Mind data?
Excellent question. I am actually part of Project Mind. I'm the director of Project Mind USA. The issue actually comes down to the privacy law, the European privacy laws. Technically speaking, no whole genome sequencing that's been done on someone from Europe can be on American soil. Now that's the rules in Europe, but America doesn't follow those rules. So, it still would be nice to get that data and make it more accessible to everybody. There are ways around these laws, even going back and getting the informed consent from the patients in Europe. You're completely allowed to do that, and you're allowed to release it any way you want. So, my brief answer is yes, we would love to have all of that data. It's just a matter of dealing with
Can you
some
wipe my
situations.
eyes?
Thank you, Dr. Landers.
So the next question is, it's actually a person with, say, sporadic ALS FTD, and they've already gotten their whole genome sequenced. Can they contribute data to the project just as individual?
Absolutely. We can, if it's a clinician with the United States, there's a good chance we've collaborated with them in the past. And yes, you can just tell them to contribute that data to us. Or just have them contact me or Dr. Jonathan Glass to have that data transferred over. It's also possible that if it's necessary to resequence your genome, which we would prefer not to do, if the funds are available, we'll do that as well.
Thank you.
So we have a question, and I hope I'm asking it correctly. But samples that you would pull from multiple entities, and then it's anonymized.
Yep.
Do you think this would lead to duplication of efforts?
Duplication of samples, or which I think is what they're getting to. That does happen often, in that ALS patients will go see different clinicians, and their genetic data will be analyzed by each one of them. And you may have one being contributed to Answer ALS and one contributed to Project Mine. And so when we get them together, is that a duplication? Yes, it is. If you looked very, very closely at those QC steps that I presented on an earlier slide, one of them is to actually look for duplicate samples or related samples. So, and if we see a duplicate sample, frankly, we just remove one of them and go on from there.
Thank you.
So this is a question on collaboration. Will all the papers that's using this data be compiled in a central location so we can know what has been done with the data? And will they be posted for preprint sites so people will have access early?
No, that's an excellent idea, actually, and something we haven't thought about. I think we've been more focused on just getting the data out there, but we can put in a requirement that upon publishing, you have to acknowledge that you got the data from ALS Compute and send us a copy. And if that's the case, then yeah, what we'll do is put together on a website that we're developing now and make it available to everyone.
Thank you.
This one's a little near to me, since I work at the VA as a nurse practitioner. Is the DoD and VA participating in efforts to give more people with ALS the opportunity to participate in donating their samples?
I do not know the answer to that, to be honest. I don't deal with the VA or DoD directly. I know there's definitely clinicians that work a lot closely with each of those organizations that this would be a better question for, but yeah, I can't come back to you, but I will contact people to find out what is going on with the VA and DoD and see if we can bring them into the mix as well.
Wonderful, thank you.
And I guess the next question really relates to that, saying it took 3 requests to get genetic testing through the VA without having familial ALS. How can we get better at this universal genetic testing? But I think what I hear is that, VA is not part of the compute project yet.
No, they're not, but it sounds like it's almost like a different problem of getting a diagnosis as quickly as possible. Some groups do it very well, very quickly, and other groups don't do it as well. And I'm sure if all of you spoke to one another, I'm sure you're going to get a different experience from every single person.
Yes. Thank you.
Thank you, Dr. Landers. So this question, do you also look for variants in other genes that appear to slow the onset or progression of people with known familial ALS who either don't get sick or who get sick a lot later than, so they get diagnosed a lot later than expected, and or have a much milder progression of the illness?
Tremendous question. The answer is yes. We definitely want to look for anything that is influencing progression. One of the difficulties I will say is that if you're looking at more of a rare mutation, it becomes a lot more difficult because there's so few samples. So a lot of these analyses, as I said earlier, are restricted to say C9 or SOD1. We are also trying to come up with other ways to look at this. I'll give you one example that we're working on with Dr. Glass. Is he has two patients that are twins, and one of them is a slow progressor and one's a fast progressor. And why is that the case? And one thing you should also know is that when we say twins, you may think they're 100% genetically identical. They're actually not. There's a handful of changes that are different between twins. And so we're going at this in terms of maybe one of those small differences may be contributing to that faster, slow progression. That's just one example. And there's many more creative ideas that are out there to kind of get to this question.
Fascinating. Thank you.
So the next question was, because people with ALS get that VUS, variants of unknown significance, do you believe that compute project will be able to make an impact in knowing the variant of unknown significance?
Yes and no. There is one committee out there led by Dr. Matt Harms, who I believe spoke to you in the past, that has formed a committee of about 30 different ALS geneticists around the world to, first of all, to go through and look at every single ALS gene that's ever been published and get kind of a consensus vote of what's the evidence that's really supporting this. Is it very good evidence? Is it not very good at all? And earlier said, we don't always agree with what's on the test panels. And the hope of this is to get it to the level of, all right, these genes are real ALS genes. These were on the border on, and these are definitely not real. Beyond that, for the ones that are in that higher classification, the next step will be to now analyze each one of those individually and say, all right, if they have this mutation, it's definitely a causative mutation. Could be a mild mutation, could be, unfortunately, a VUS, as you stated, but that is kind of the next step of this particular committee to take it that much further. One thing that ALS Compute hopefully will be able to do, though, is to contribute to say, all right, this is how many people we see with this particular mutation and how often we see it within ALS patients and how often we see it in the general population. And that if the numbers are large enough, which is what you're usually trying to get to with genetics, you want larger numbers, then hopefully that will increase the evidence for either a particular gene or a particular mutation being confirmed as being, authentic.
Thank you. There were a couple of comments, then questions I wanted to. So the comment was, it would be great if there's a central portal or a web page where people can actually know how to upload their genetics. So either they can contact, so that would be giving people some direction. And also there was some comment on someone here as actually doing analysis and also would like to pull out individual VCF results by GUID, is it okay if we can actually have that person contact you directly to discuss it?
Absolutely. Yep.
Okay, perfect. So with that, Dr. Landers, thank you so much for giving your hour, and we
Pleasure.
really appreciate it. And we had, hundreds of people here, and we all took away something today to feel hopeful. So thank you so much. And we'll say good night to you, and we actually open up the forum for the rest of the people to discuss among each other.
Thank you very much. It's been a pleasure, and keep up the good work.
Thank you.
Thank you.
Bye-bye.
Folks, I keep telling you this. Look what came into your living room tonight. This is why you subscribe and join your extended family, because we get to see each other twice, 3 times. This is a community, and we are the core, and we are actually detectives. So, the doctors have been working on ALS for a long time. They might need some help, and they might need it from us. And so if any of you would like to help contribute some of your time, energy, you can always contact us and say, what can I do to help end ALS? Because together we can do it. And so I just want to say, if any of you have time to join any of our projects, we are really working to find out what this is about, how it can be ended, and how we can be involved in the answer. And more than anything else, I think each one of you are part of the answer. So
absolutely. Even just getting together for these meetings once every week or two, it's such a healing thing to get that positive influx and hear your words and everyone else's words. It's all good medicine.
Thank you so much, Sean. This is exactly why we're doing what we're doing. So this means a lot to us.
Yeah, well, we thank you, Indu, for creating this little monster of yours.
Well, big smile, everybody. Come on, give it to me, please, just as much as you can. Thank you so much. It's really a power. And it was Teresa. One day I said, let's say goodnight, everybody. And Teresa wrote me a note, and she says, McFinn, I can't speak, but I can make a heart. And so I'll always remember Teresa coming up with the heart. Yeah, thank you. Thank you, Karen and Patty and Ruth. I mean, and Dee Dee, these are little symbols. Eric,
If you have anyone that you want to have us invited to speak, please write to us, info@everythingals or McFinn or myself. We would love to bring someone, that you are interested in hearing from. And if there is any feedback for us that we can do better, feel free to let us know as well, because we're doing it all together. And the more feedback and more, we can get involvement, the better we all become.
Great. Let's all say goodnight to each other.
Good night, everybody. Good night, Indu.
Good
Good
night.
night, Ruth, Patty, Chuck,
Night, everybody.
Little Peach.
Good night.
Love you, Paul, Kelly, Janie Gobo,
everybody.
Belly. Thank you, Janie.
Bye,
Here,
Sean.
yes, Faith, I'm sending it right back to you.
Hang in there.
Chuck Shaw, aloha to you, Chuck.
Oh, look at that heart, Mary and Teresa.
Right, and Eric, my man who makes people happy.
Good night,
Thank you
Mary.
very much.
And the puppy there.
I can.
Right, Brian, Kelly, Marie. All right, Bob, Eric Brown. Love you, Eric. And Mary, thank you for having
Good
this
night,
angel
Mary.
right next
Thank
to
you.
you.
I
He
love
knows
it.
how to make parts. It's really good.
Good night.
Bye, everybody.
Bye.
Bye-bye.
Bye.